Okay, so we go to the last speaker of the day of the events as well. So the next talk is going to be given by Greg Sands. So Greg is a senior research fellow at the Auckland-Boston Institute, and he also did his PhD in the University of Auckland, so in the engineering science department. His focus has been on the structure and electrical function of the heart, beginning with computational models of electrophysiology, moving through experimental and clinical measurements of cardiac activation to develop of imaging systems for understanding tissue structure organization in health and disease. And I think this talk is going to be about the MµSIC. So the word is with you.
Thank you everyone for sticking around. This is the last talk of the workshop, as Gonzalo said. So, and thank you to Gonzalo as well for inviting me to talk. I was a little confused when he did ask me, because despite the title of the workshop, I don't really do AI, and I don't do medical either, but the overlap seems to be in imaging. And in fact, in a couple of the other talks, especially today, there are definite areas of overlap. So I hope even if you're mainly interested in the AI and medical parts, you'll find some things that are of interest. So I gather this workshop is a little bit about kind of meeting and seeing other people as well. So I thought I would give a very, very quick overview of my history, which has come from somewhere well outside anything even to do with imaging at all. Started off with a PhD in computational electrophysiology, so looking at electrical activation in the heart and writing a computer model of that accounts for a moving geometry. And then moved on to experimental work, building plunge needle electrodes and recording electrical activity within the heart, and then reconstructing that relative to the structure that lies underneath. A little bit of clinical work for a time, measuring electrical potentials on the body surface and using inverse methods to reconstruct what's happening in the heart. And we did actually use some echo in that in terms of trying to determine the heart position and shape. But that was very, very early 2D echo with a magnetic probe that tried to align a few different images from the same probe, well before these lovely 3D echo sort of images are available. And that moved on into microstructural imaging. So underlying all of these things to do with computational models and experimental measures and clinical models is what's actually going on in the heart itself. What does the tissue look like? How can we understand the structure and how that relates to the function of the heart? And that kind of gets us to where the interest is in this workshop, which is in the range of scales that you need to consider when you're looking at any organ. For me, I'll talk about the heart, but we can apply the same thing in the brain and many others. And it spans many orders of magnitude in terms of scale, from tens of millimeters for the whole organ, right down to nanometer resolution when you're looking at DNA or cell membranes or individual proteins that make it up. And we want to have different ways of looking at this data across perhaps some or all of these range of scales. And a lot of the medical imaging techniques that have been talked about, they work up at the top end. You're sort of from 100 millimeter size and maybe 1 millimeter down to maybe 100 micron resolution. What I'm interested in is kind of a slightly broader range that still covers larger tissue sizes, but extending the scales down into the micron range. So when we look at that, the underlying goal behind this is determining something about the quantity and the relationship of microstructural features within a tissue or an organ over macroscopic dimensions, and doing so in 3D. And we've heard some, and there are a lot of different approaches that you can use in order to do this. And they all work well in some ways, and they all have some constraints. So just out of some of these, we have confocal microscopy, but it's typically slow. You're limited by your sample size. Light sheet microscopy, again, new advances are getting much better, but the resolution is still somewhat compromised. MicroCT, MRIs, and so on. So identifying particular structures within the tissue. Can you tag things or label particular elements in a way that that make them stand out? You're relying on intrinsic properties like the density in terms of X-ray imaging, or the water content in terms of MRI. Histology, I might have to rewrite some of this because what was shown today about realigning histological images looks to make this much more practical, but it's a inherently very huge amount of processing and manual work required in order to put all of these individual sections together. And then multiphoton has also been mentioned, and again, a big issue to do with that is the speed of acquisition. And when it comes to looking in 3D at things, then we also need a way to optically section. If we're looking at optical methods for looking at tissue, we need to be able to optically section our sample. If we just have a wide field image, then yes, we see an image plane where things are in focus, but all of the planes in which things are out of focus are also acquired by the detector to some extent. And so we have a blurred image that results. This is what you do with histology. You slice it up into individual sections, and that constrains your optical plane. But you have the problems of re-registering all of those, realigning them and stacking them back into a volume. Again, maybe some new techniques are making that a little bit easier to do. Light sheet's been mentioned a lot, and in this case, we construct an optical section by shining a beam of light perpendicular to the direction that we're imaging. And so we only, if you like, illuminate a single plane, hopefully a fairly thin plane through our tissue. And so we only get image data from that plane. And then as we move that around, we can build up our three-dimensional image. But there are constraints on this in terms of shadowing, in terms of needing two orthogonal planes in which to illuminate and to image. And there are resolution constraints, especially out of the center of this light sheet. And then confocal is a method that's been used for a long time, in which we optically section by rejecting all of the out of focus light. I'll go over this a little bit because this is an approach that we've developed a little bit further. But the inherent problem with confocal microscopy is it's essentially a point-by-point approach, and it has been slow and difficult to use for large samples. So just in terms of confocal microscopy as it's used for fluorescent optical imaging, the basic way in which it works is that we tag some particle with a fluorescent probe or fluorescent dye that attaches just to that, maybe a protein or a membrane, and it receives some excitation light and then emits fluorescence at a slightly different wavelength. And that allows us to identify just a particular thing that has been tagged within the tissue. And then in confocal microscopy, we use a pinhole that rejects light that comes from out of that focal point. So using a laser, we can construct a beam of light that gets focused down to a fairly small point within the tissue in the microns sort of size range. And then with a pinhole at our detector, we can reject all of the light on the right-hand side there that arises from somewhere that's out of focus, and only, as on the left, what the fluorescence that comes from the point that is in focus of that light gets recorded by our detector. And then we need some way to scan that point around our tissue. And usually that's done with some galvos that sweep the light across, stages that move the sample around underneath, and so on. But at its core level, confocal has usually been a pointwise approach and relatively slow. Nonetheless, we spend quite a number of years developing systems that would extend a pointwise approach into larger samples, but that in the end proves too slow once we want to try an image over the millimeter kind of range, but keep it at the micron level. And so that's led to the design and the construction of a home-built microscope for doing imaging. It's called MµSIC, which is an acronym that stands for macro microstructural imaging with clearing. And so what we want to do is combine tissue clearing techniques that make tissue relatively transparent to light with a scanned line confocal microscope, and I'll go over what that is in a minute, to enable microscopic imaging of macroscopic volumes of tissue. We have a number of components that make it up. The most important one is probably the objective lens, which allows us to focus the light into the sample, the camera that acquires the images, a large RAID array that allows us to store these images that arrive very, very quickly and end up being very large, stages to move samples around, laser for excitation, and an FPGA system for controlling it all and integrating the control and the data acquisition in the process. The basic optical design is actually a very, very simple microscope. We just have a laser coming through a fiber. It's aligned, and then the beam's expanded up into sort of a 1-inch beam. And then that's passed through a cylindrical lens, and that focuses the light just in one axis. And so we end up with a focused line of light. Instead of a point, which would be the normal case in a standard confocal, we form a line of light and then image that line through the objective lens onto our sample. So this illuminates a line of the tissue at a particular depth, the focal depth of the lens. And then the fluorescence arising from that line is transmitted back through the objective lens and onto a single line of a camera. So we acquire, instead of one point at a time, a whole line of pixels at a time. And then we move our sample under that line so we can build up an image strip, and then translate the sample to produce an array of strips in 2D and then in 3D. And by doing this, build up a complete image volume over the order of tens of millimeters of size or thereabouts. And so this microscope has a voxel resolution in the sub-micron range. We can sample it at a 1/4 micron, but the true resolution of the lens is just a bit under 1 micron in plane. We're acquiring lines at up to 25,000 lines a second. So that gets very large, where it can be acquiring a terabyte of data per day. Up to 4 fluorophores, 4 lasers are in the system. Due to the objective lens that we have, the 405 doesn't work so well, so we have essentially 3 imaging wavelengths. And then sample volumes with the system that it's designed at the moment is currently up to about 25 millimeters square. And if we've got tissue we can see through well, up to 8 millimeters in depth. And really that is the thing. Can we see into tissue well enough that we can image whole blocks of tissue at least half or 1 millimeter or more in depth? And this is where the tissue clearing process comes in. This is a process of making tissue essentially transparent, or as transparent as possible, through permeabilizing cell membranes, get rid of the fat that's there and decoloring the tissue so we're not scattering light, we're not absorbing light, and then matching refractive index so that we get a uniform entry of the light and the fluorescence detection through the sample. Now this method has been around for about 15, 20 years or so. There are now at least 40, probably way more than that, different methods for doing so, often targeted to quite particular things, but a lot of them have been developed for brain tissue, and brain seems to be very well adapted to clearing. And these methods allow us to have optical depths in the range of a couple of millimeters through to potentially 8 millimeters or so. So I want to just go on and give a few examples of the sort of tissues that we've been able to image using this, and the ways in which, the sort of images that can be created, but then the questions that arise from that data and what might be able to be done. And that's the sort of area where some of the AI methods that have been talked about may be able to play a part. So this first one is in terms of the vascular, the coronary vasculature within hearts. Looking at whole rat hearts, we clear them using a perfusion process, so perfusing the clearing solutions through the tissue and making the heart essentially transparent, so that if we look at it on paper here, it's clear rather than red and not able to, and opaque. The vasculature has been labelled with a lectin that's perfused through it. And then doing that, we can acquire images of whole short axis sections of this heart at around 1 micron resolution, but over tens of millimeters in size and about 1 millimeter in depth. And this is just a maximum intensity projection of through some of that. And then the sort of 3D vasculature that's able to be extracted from this image. Just showing a portion of that, and this is a little video that Vibu, who was the PhD student who did a lot of the analysis work for this put together. So showing the high resolution, but over the large scale, and then here the process of reconstructing that network. So extracting out blocks of that data, doing image processing, deconvolution and denoising, some filtering, thresholding and segmentation, and then centerline extraction, all in 3D space, so that what we're coming out with is a 3D network of the coronary vasculature at capillary level. So where these vessels are on the scale of 4 to 10 microns diameter, but over extended regions of the whole heart. There's a second example, and one where a lot of the funding for this work came from, was looking at the right ventricular outflow tract in humans, and particular questions about the way that gap junctions, which are an important part of the electrical activation process, the way that they change or how that varies across quite a large scale piece of tissue in the right ventricular outflow tract. For this one, we had access to a human heart, which was provided in a collaboration with researchers in Bordeaux. And for clearing this heart tissue, human heart tissue is relatively difficult to clear. A lot of the established techniques don't really work particularly well, or at least we need to augment them in several ways. And for these, we combine multiple clearing approaches. One is methyl salicylate, which is a dehydration approach that works extremely well for fatty tissue. Cubic, which is a very standard approach for a lot of different tissues, which does well at clearing the musculature. And we see on the right here the tissue as it ends up at the end of the clearing process. And then we're able to label parts of this tissue with different labels. These ones here, so the green is WGA, it stains a lot of the cell membranes, so a lot of the connective tissue, the vessels within the sample, and NAV 1.5, which is a sodium channel. And so the image that's shown here is nearly 20 millimeters long, 4 millimeters high, and imaged at 0.5 micron resolution. And right through, and I think the depth of this was about 350 microns. And across this whole image volume, we're able to see the fine structural details of the individual myocytes and the vessels that surround them. And the sodium label is telling us where there are the sodium channels are present within the cell membranes and adjoining individual cells. We can also look at the gap junctions a bit more directly using connexin 43. This is a label that is localized to the gap junctions, and again see this high level of detail across large 3D images. So within this, we have a 3D image volume. This is just extracted out a 1 millimeter square by 300 microns deep sub-region of the full image, but the full, this same level of detail is present throughout the whole volume. And as well as identifying just the image as, visualizing the image like this here, this was also some work that was done to pull the coronary vasculature out of this, similar to what Gail was talking about yesterday, except instead of an AI approach, it was the Frangi filter that he was talking about, which identifies the structures within the image that have vessel-like properties. So they're long and thin and connected in this kind of a network. And then we can also extract the information about that is useful for computational modeling, and this is work that Mark True has done a lot of work in. So we see the fiber orientation in 3D very easily pulled out from these images using a structure tensor type approach. And then from the image of the gap junctions, we can quantify the distribution of those, the conductivity of those, and create representative tissue units that can then inform a model of the electrical activity through the structure that has been imaged from that particular heart, or that can be used to inform more generic models of electrical activation. And the third example, and this is more recent work, is looking at ganglionated plexi on of the intrinsic cardiac nervous system. And these are the nerves that arrive into the atria at the top of the heart. And we have access through the hospital to human atrial tissue collected from patients with and without atrial fibrillation who are undergoing a coronary artery bypass graft. And in this case, we're interested in imaging the neurons and the cells, soma, and the plexi that arise on the surface of the heart, and looking at the differences between atrial fibrillation and non-AF patients. Again, we're able to clear this tissue reasonably well. So most of it is fat. This is extracted from the fat pads that lie on the surfaces of the heart, and the nerves are embedded in these. And using the methyl salicylate especially, we're able to remove most of this fat that would block any light coming through the tissue. And then this is a scout image from that sample, and we can see 4 highlighted here, different ganglionated plexi that are on the surface of this heart. And then from this scout image, then we can do a higher resolution 3D image of just some region of this. And so if we just look at this here, we have a single GP with the nerves that are coming in, the individual cells that are here, the ones that are cholinergic, noradrenergic, or a mixed expression of both. This is just for 2 labels. We've also done some that also look at synapsin, which is the connection between the nerve and the cell, and the distribution of those in these regions. Now, I just want to finish with a few comments about some of the issues related to doing all of this. One of them is speed. So in order to acquire large 3D images of large volumes, you need to do it quickly or you're waiting around a long time. And so this technique is bringing data in at about 500 megabytes per second. This is extremely fast. We can store this onto an SSD, but an SSD is going to fill up very quickly. You're going to have a couple of minutes of imaging and it'll be full. And so, or maybe a little bit more, but using a large RAID, we're able to parallelize that storage of data across a number of disks and image at this rate over a reasonably long period of time. The size of the data is also an issue. These images are very large, from tens of gigabytes up to several terabytes for a single three-dimensional image. Often it's far too big to fit in memory, too big to fit in a GPU or even a standard computer memory. There's a lot of things that need to be done in order to handle images of this size. One is in storage formats. So rather than using just a standard, say a TIFF file or something that puts all of your image data into one container and makes for one very big file that's difficult to handle, to look for formats that allow for a hierarchical and chunked storage that store parts of the image in individual places. So you only need to read in what you need at any one time. Using, for the computational and image analysis, you really need to use something that can split up your problem into multiple parallel units, either to solve sequentially or to solve, if possible, at the same time. Dask is a good example of a Python library that kind of transparently allows for this, so that an algorithm that's written in NumPy can be quickly transferred to work on a chunk-by-chunk basis, and according to the capabilities of the machine that you're running on, to choose the way in which that is done. And also to use multiscale viewing software. Napari is one example that we've used, NeuroGlancer, and there are several others that allow for a quick, sort of like a Google Maps type viewing interface to this large 3D data. So when you want to see the whole thing, you only load up a low resolution of your image and can kind of orient yourself, work your way around. As you zoom in on particular regions or crop the image down to smaller regions, then you load in more of the high resolution data into that environment. And the third point I want to make is just getting the most out of the data. So inherently, optical systems have a blurring of the images. I'll just go through a couple of these things in a minute. But one of the ways in which we can increase the resolution, or the way that our image represents the true case, is through using deconvolution to undo some of that blurring that occurs as part of the imaging process. So just a couple of slides about these kinds of things. On the left, there's a difference between a kind of the traditional monolithic file format, a TIFF file, where everything sits in one file, and to extract a portion of it, you've kind of got to know where to, you know, if you can load part of the file, where you need to go and where you bring that in. ZAR is a fairly new file format, although the Open Microscopy Group has been accepting it now as one of their next generation file formats. It's really designed for very large 3D images. And it breaks the image down into individual chunks that are stored separately, and allows, almost like the tiles in Google Maps, as I said, and allows for that to be flexibly read in. And not just the individual tiles that are stored, but at multiple scales as well. Then Dask is the Python library that essentially takes a large image or a data structure which may be too big for the memory of the machine that you're working on, but transparently breaks that down into smaller chunks, and will allow you to write code that processes through those one at a time, maybe doing some in parallel where it can. You need to pair this with algorithms that work for chunk type processing. So filters work pretty well. A lot of the deconvolution image processing filters. They're suitable for doing on a chunk by chunk basis, as long as you're aware of the boundaries between chunks and making sure that you don't introduce abnormalities there. But harder things are things like constructing networks or graphs from your data, how you connect, if you're processing individual chunks, how do you connect up between those chunks well? And so those algorithms need a lot more thought, or they become very difficult to do in this environment. And then this is the sort of interface we have with Nipari, where we can have a large multi-color 3D image that we zoom in on parts of and can view at different scales. Deconvolution, just the last thing I want to talk about. We'd love it if, when we focused our light, say, to a point, that we could make that point as small as the voxel that we acquire, and all of the light would focus on that point, which means all the fluorescence would come from that volume, and that's all we would see. The reality is that with any optical system, that point is actually spread out across a larger region than a single voxel. And this is what's known as the point spread function of the microscope. This is a measurement of the point spread function of the MµSIC system that we have, and that was acquired by imaging microspheres, so spheres that are 200 nanometers smaller than our pixel resolution, or even our theoretical resolution of the objective lens, but the way in which that light is spread out. What that means is the light from any one point is spread out over a whole number of voxels of our image. Converse of that is that any voxel that we've acquired contains the light from a number of different points, of true points within our tissue. What we'd really like to do is recover the true object by kind of inverting this deconvolution, which is deconvolution. And so we use this point spread function to, we kind of use an iterative, maybe somewhat regularized algorithm that reconstructs an image that doesn't have this, or has as little of this blurring in it as possible. And the difference that it makes is quite substantial. And I'll just show it with a pair of images from our stack. On the left is a raw image where there's a lot of out of focus light, and the structures are blurred. It's much harder to see individual fibers, whereas on the right hand side, we can start to resolve some of these structures. These are nerve fibers within the tissue, so these are smaller than the optical resolution of our microscope. That's why we can't quite resolve each one independently relative to each other, but it's a huge improvement in all three dimensions on the raw image that was acquired. So if we go back, this was the goal that we had in starting this, and the sort of system that we wanted. And for the most part, we've been able to achieve a lot of this for the kind of tissues that we're interested in. Most of our work at the moment has looked at, so far, at things related to the heart, but a range of different features: vasculature, cellular organization, gap junctions, nerves and GPs. We've also done a little bit of imaging of lymph nodes, of carotid bodies, and in the future, are planning to extend this in a number of different directions, looking at imaging of the stomach, of the uterus, of the lymphatic system, of the brain, and potentially any number of other things that this can be applied to. So let me just finish with some acknowledgements. There's probably 3 times as many people that aren't on here, but these are the main ones that contributed to the images and the work that was shown, both here at the ABI, at the physiology group up the road, and collaborators in Bordeaux, and also funding primarily for the system from the Leduc Foundation through a Transatlantic Network of Excellence grant and the Royal Society in New Zealand. Thank you.
Questions?
Great talk. Love it. And if you may, I'll bring back the word medical into the.
Yeah.
So my question is, have you, I think, and it's with regard, it's kind of like related to the last example that you showed with multi-structure imaging and visualization. Have you tested that on something like, let's say, meat tissue, then fat, then blood vessel with the fluid inside it, being able to reconstruct that kind of structure in microscopic scale type of thing?
So putting all of the structures together. Yeah, in fact, it was an image I left out because I thought this was getting too long, but it's something that Mark has done in terms of that layering of different structures onto each other. Yeah, absolutely. That's the end goal, is to be able to do that.
Okay.
Yeah.
That's a project.
Yeah.
One question regarding the field of view and the applicability of the system. So
Yeah.
how big can be the sample that you can scan in one go?
So we need to sit the sample in a bath of oil. So we have a uniform refractive index between the objective lens through the oil and through the sample. The baths we've made at the moment will allow for a sample up to about actually 30 millimeters square and maybe 5 to 8 millimeters high. So that's a physical limitation in terms of being able to, of the scanning region that we could do. We've had thoughts about building a bigger bath, and that would be possible. You could extend it to 100 millimeters with the stages we have, but the imaging time goes up with that. But those are the only real constraints. The larger bath could be useful in a case where you had a large piece of tissue that you wanted to focus on some subregions of, but you didn't know where they were. So you wanted to do a coarse scout of the whole sample first, identify the regions you're interested in, and then come back at higher resolution and just image those smaller regions. And that would be possible.
And there are in-house techniques for doing the co-registration problem.
Co-registration of
So when you have the small sample to automatically detect where it's going to be. Anyway, you have some data as well.
Yeah, so because everything's mounted on the stage, the tissue's fixed and embedded and mounted there, then once we've acquired the scout image, we know exactly where we need to go in order to pick up those individual locations. Yeah. Yeah, Paul.
How well conditioned is the deconvolution process? Because it looked like the point spread function, covered many voxels.
Yeah.
And in a case like that, especially where you've got some noise in your measurements, that gets amplified through normal, what do you call, conditioned, I suppose, or regularized deconvolution.
So deconvolution can have a problem with noise. There are kind of two main approaches to doing it. One is an iterative technique, which has something like a Tikhonov regularization, which tries to minimize the noise. We don't use that. It has some advantages, but it is very difficult, I think, to get working well. The Richardson-Lucy technique is probably the most common one, and there you start with either a flat field image or perhaps a blurred version of what you've acquired, so something that has the low resolution spatial information. And then you convolve that starting image with your point spread function and look at the difference between the result and the image that you collected. That gives you a correction factor to make to this, this image. And then you sequentially apply that again and again. And so because you're starting with a blurred image, the noise really only comes in after a large number of iterations. So you need to choose a point in which to stop that process. So you have recovered the detail before you start recovering the noise. Essentially, I guess it's working through it's adding higher and higher frequencies as it goes. Yeah, in practice, we will see some noise amplified, and there's some inherent noise in the imaging system that we have to be careful not to amplify, and because it will dominate the image. So yeah, it's an important point.
Also regarding the point spread function, all the wavelengths will have the same point spread function?
No, that's very similar, but they are different. So we've measured it separately for each wavelength. These microspheres have 4 fluorophores in them that activate at different wavelengths. So we're able to do that. Also, the imaging properties of the lens are different at different wavelengths. We got a very nice lens that would do the depth and the, had a lot of parameters that we really liked. Unfortunately, what had been designed for, and we didn't quite realize all the implications of this at the start, but had been designed for multiphoton imaging, where you're using much longer wavelengths. And that's one reason why 405 doesn't work well. And each of the wavelengths as we go down has some different chromatic aberration associated with that. But again, we can characterize that by imaging microspheres or something similar, and make that part of the image reconstruction process. So we're trying to undo as much as possible of the artifact that the microscope is introducing in its imaging.
And can you not use the information from the different optical lenses to better pose the problem of the deconvolution?
Yeah, and in essence, that's what you're doing by imaging the microspheres. You're characterizing the whole optical system. So both the excitation and the detection paths are all kind of merged together into that. And so you are, as best as you can, characterizing the microscope within that.
I might have missed it, but how big is the size of the lens, the current one?
The lens?
Yeah, like the old camera system or the light emitting system.
Trying to think what you mean. What do you mean by the size of the lens? You mean the diameter of it? So the lens itself has a diameter of about 40 millimeters, I think.
And how small could it be if you want to make it smaller? Does the current technology allow to make a very small, like let's say 5 mil lens? I know it's very high tech, but
yeah. So one of the things with this lens is it gives us a high numerical aperture.
Yeah.
So we have an NA of 1 over quite a wide range of refractive indices, and a long working distance of up to 8 millimeters. That's a very difficult thing to get in a single lens. In fact, this lens has, I think, 25 different glass elements in that, along with a correction collar that shifts them relative to each other. In that case, it wouldn't. There is another lens that if I had some money in my back pocket, I'd love to get, which has a curved surface on the front. And so that has the big advantage that the light that's coming out of that front surface is perpendicular to the glass at all points across that. It's not like a flat surface that it's entering at an angle. So that means you get no refraction at that surface, independent of whatever change in media that it's going into. And that is a slightly smaller lens with a higher numerical aperture, and it's well corrected across all the wavelengths. That'd be probably a much nicer one to have. But I still don't think you'd get that down to a few millimeters in size for this sort of thing. Yeah.
Hi, Greg, can you hear me?
Yeah.
Hey, this is really awesome because it gives us a really good understanding of geometry and composition of these tissues. I was wondering, what do you think will be necessary to start looking at, added information, for example, mechanical properties of these? of these tissues that are being imaged?
I guess it would require mechanical experiments to be done first. You would have to do that in living or, you know, tissue that you'd kept in a viable state. There's a lot of work on at the ABI here on doing that. But then I guess once you have characterized that, you're able to clear the tissue and image it in this way. So those hearts that I was talking about from Bordeaux, and I didn't cover this at all, but the original, where they came from originally is that the researchers were doing optical mapping of electrical activation on the surface of those hearts, and then have taken MRI of that, and then we've been able to extract a part of that and do this this imaging. Our hope is to do the reconstruction back. So we take that structure back into the MRI and then translate that back into the optical imaging of it, optical mapping of activation on the surface, and relate the structure directly to the experimental measurement. And I think some of the techniques that were talked about earlier today, such as the tensor reconstruction tool, something like that is going to be really powerful in being able to allow for that to happen.
Question? Let's keep another one close. Okay, and that was the last presentation of the event. So thank you very much for showing, for being able to connect online to the workshop and the people that are in person as well. And please send us email to me or Sean if you want to contact any other of the participants and want to establish a collaboration. Thank you very much.